Assalamu alaikum students, this is your biology class and our today's topic is excretion in man. This is the third topic of your first chapter, homeostasis, and I am your teacher, Miss Hira. First of all, excretion in man. The main excretory system in human is the urinary system. Actually, the excretion mainly done by urinary system, but some of the excretory products are also removed as sweat or as tears and also from liver. So these are the other organs. First, urinary system is the major excretory system. Lungs exhale carbon dioxide as the excretory product. Skin remove sweat from the sweat glands, as well as liver perform very much important function in whole body metabolic activities, and also perform excretory function as it produces bile. Liver is called important homeostatic organ. Why it is called homeostatic organ? Because it performs major functions of homeostasis or balancing in the body. As you see the picture, in which liver has two lobes. One lobe is larger and one lobe is smaller, and in between these lobes ligaments are present which attach these lobes together. And here is a green ball like structure which is called gallbladder. It collects bile from liver and extract it and excrete it in the small intestine. Here are the homeostatic functions of liver. First, metabolism of carbohydrates and lipids, deamination and urea formation, production of bile, detoxification, formation of cholesterol, thermoregulation, storage of vitamins. First, metabolism of carbohydrates and lipids. First, the carbohydrates. Prime importance in carbohydrate and lipid metabolism is that 30 to 60% of the indigested carbohydrates are taken up by the liver and stored as glycogen. As we all know that glycogen is the form of carbohydrate and it is the stored form of carbohydrate. When glucose need to be stored in the body, it do not store in the form of glucose, but it convert itself into glycogen and then store in the liver. In the fasted state, the liver release glucose by glycogenolysis and gluconeogenesis. These are the two processes of conversion of glycogen to glucose. The liver therefore act as a buffering organ in carbohydrate metabolism to keep the blood glucose level in a physiological state. We have already discussed this example in negative feedback, so you can also touch that topic for this reference. Next is the metabolism of lipids. The liver produces cholesterol, triglyceride, phospholipid, secrete and uptake lipoprotein, and discharge cholesterol in the form of bile. It actually metabolize lipid in different forms or different types of lipids. In addition, liver produces important enzyme for lipoprotein metabolism. It also produces enzyme. In consequence, many typical features are observed in lipid metabolism in hepatic diseases. So liver help in carbohydrate and lipid metabolism in many ways. Second is deamination and urea formation. Urea formation is the easy word, formation of urea. And deamination means the removal of amino group from any molecule. So this function is major and performed by liver in situations of excess protein intake. When there is much protein present in the body, they need to break their amino acid for energy. Okay, so amino group is removed and converted to ammonia with the help of liver. Here is the equation. This equation is just for your knowledge. Otherwise, it is not necessary according to your content in your book. Ornithine cycle. Ornithine cycle is actually the formation of urea, but how? First, deamination occur, then from the removal of ammonia, that ammonia combines with carbon dioxide and form carbonyl phosphate that convert to the citrulline, then arginosuccinate, then arginine, then ornithine. The product which recycle in the cycle is ornithine. That is why the cycle named is ornithine cycle. And the product after arginine, if the product become ended, then the end product will become urea. That is why the cycle is also called urea cycle. At the end, the product will form is urea, but when urea do not form and the cycle goes on, then the product will become ornithine. So two products are there. The end product is urea and the recycled product is ornithine. These two names are given to the cycle on the basis of the end product. You have to just read the name and need to draw the cycle for your board exam. And you have to learn the name citrulline, arginosuccinate, arginine, and ornithine, and urea only. Okay, now the third function is production of bile. Bile is actually the end product or excretory product which is green to yellowish in color and produce and store in the gallbladder and help in the emulsification of fat during the digestion in the small intestine. Bile is produced continuously by the liver and is stored in the concentrated form in the gallbladder. So bile is produced by liver. Now detoxification and elimination. Detoxification means removal of toxic substances from the body. Elimination means excretion or removal when toxic substance is detected and eliminated by the liver. From lungs, from skin, from gut or intestine. Liver detoxify all the organs and then toxic exit through skin, by sweat, kidney, by kidney through urine, by colon through rectum, bile के through करता है, and lungs through lungs in the form of carbon dioxide. So toxic stored excess toxins get stored in fat cells, bone marrow, liver, central nervous system, brain. Side effects of toxicity maybe can cause. These are all the side effects which are caused by the liver abnormalities. Joints, blood, tissues, muscles are also the site where toxic substances are present. So liver detoxify all the organs of your body and prevent these all the effects like autoimmune disorders, joint pain, arthritis, allergies, acne, headache, obesity, overweight, cardiovascular diseases, chronic fatigue, hormone imbalance, and so many other diseases. Next is formation of cholesterol. Formation of cholesterol, how cholesterol is formed? The liver synthesizes cholesterol from acetyl-CoA. Acetyl-CoA is the end product of glycolysis. Then cholesterol pool in liver cell also receive the dietary cholesterol, which is contained in the chylomicron remnants that are formed through the extraction of triglycerol from chylomicron by lipoprotein lipase. This is the whole process of making of cholesterol. Cholesterol is actually is of two types: good cholesterol and bad cholesterol. Bad cholesterol never be formed; it always is excreted from the body, and good cholesterol have to be formed, and liver help in the formation of good cholesterol. Next is thermoregulation. What is thermoregulation? Thermoregulation is the homeostatic process and it is done by so many organs: liver, brain, heart, skeletal muscles, all are involved. Liver play important role in the detection and balancing of temperature throughout the body. And the last one is storage of vitamins. Storage. Vitamins are naturally present in our body, and vitamin A, B, and D can be stored and are stored in liver. That is why we can say that liver is the major or important homeostatic organ and have individual importance throughout the body. As you know that liver is the largest gland of your body, so it causes so many secretions like bile. Now our next topic is excretory system. Excretory system in man consists of in excretory system we will focus on urinary system. This is the excretory system of your body where two kidneys are present, and from these two kidneys tube-like vessels arises which are called ureter, and two ureters combine and merge in a sac-like bladder which is called urinary bladder, where urine will store in your body, and then when this bag is full, then excrete out from your body through the anal opening. Now, closer structure of kidney. Kidney actually is present in left and right side of the body. Left kidney is upper and right kidney is much little lower from the left kidney. Both kidneys have on the upper end, both kidneys have glands, and the glands which are present on the upper end of the kidney are called adrenal gland. Two vessels, blood vessels, artery and veins, both enter and exit the kidney. Our blood vessel connected to the kidney are termed as renal. So we can say that renal artery, renal vein. And this is the tube which is called ureter. Okay. After that, kidney in the human body. What is the structure? What is the size? There are two kidneys situated in the upper part of the abdominal cavity, lower the diaphragm, one at each side of the vertebral column. Length of the each kidney is about 12 centimeter long and about 7 centimeter wide, while its thickness is about 3 centimeter. This is the closest and internal structure of kidney. As you know that kidney enclosed, all the internal organs enclosed in particular type of membrane. So kidney enclosed in kidney has also enclosed in a particular type of membrane which is called peritoneal membrane or peritoneum, and has the upper end of kidney has adrenal gland. When you cross section longitudinally the kidney, each kidney has two major portions, outer portion and inner portion. The outer portion is called renal cortex, okay, and the inner portion is called renal medulla. Renal cortex is the smaller or thinner portion than the medulla. Medulla portion can be differentiated from cortex by these triangular pyramid shaped structures. These pyramid shaped structures are called renal pyramids actually, and these pyramids, all these pyramids collect tubularly and form a cavity which is called renal pelvis or pelvis cavity. Artery, renal vein and renal pelvis, these are collectively enter and exit through the area in kidney. This area is called hilus. It is written in your book. Renal pelvis arises and make a tube which is called ureter. After that, the innermost structure of kidney. Kidney has the most innermost structure and millions of microscopic tubules which are called nephron. We have already discussed the term protonephridia, metanephridia, nephrostome, in which I have already told you that nephron, nephra, nephridia, nephro are the terms related to excretion. So we can say that the nephron is the finally complex, most complex structure of all these structures on the evolutionary point of view. It is the functional unit of kidney. We can say that it is the functional unit of kidney as well as a structural unit of kidney. Each kidney has millions of nephron in it, which perform osmoregulation as well as excretion. As you can see that this is the smallest portion of kidney which is zoomed in this section of the diagram. Here you can see that all the yellow portion, these tubules, all the yellow portion of these tubules are actually the nephron, and nephron is also have a bunch of blood capillaries, so that we can say that blood on the capillary level filtered in nephron, and making of urine is possible by the nephron functions. Now the nephron. Nephron is actually two major portions. Nephron actually have two major sections. One is a tubular structure and the other is blood capillary section. So first we will discuss the tubular structure. Tubular structure has a spherical capsule, C-shaped capsule or cup-shaped capsule, which is called Malpighian corpuscles or Malpighian body. This capsule consists of two sections. One is Bowman capsule. The tubular section of this is called Bowman capsule, and this capsule or cup have capillary network in it to filter, which is called glomerulus. Okay, glomerulus have two entry and exit point, afferent arteriole and efferent arteriole, but the grape-like bunch is called glomerulus and Bowman capsule. This section is completely called Malpighian corpuscle. After that, this Malpighian corpuscle's Bowman capsule raise and form thick tube-like structure which is called proximal convoluted tubule. Proximal means before, convoluted tubules, folded tubules. Okay. After these proximal convoluted tubules, a loop is present which is called loop of Henle. This loop have two sections. One is downward. Downward loop is called descending loop, and one is upward, which is called ascending loop. After that, just like proximal convoluted tubule, another section of tubule is present which is called distal convoluted tubule. After that distal convoluted tubule, whole nephron is connected to a thick major duct which is called collecting duct. This duct is actually connected with 4 to 5 or 5 to 7 nephron together. Each nephron make a drop of urine which is collected through the distal convoluted tubule to collecting duct, and this collecting duct give the urine to the pelvis cavity, and from pelvis cavity this urine will go to the ureter. After that, the yellow portion is the structure which we have already discussed. Here I am also repeating here. This is the section which is called Malpighian tubule, in which capillary network is called glomerulus and the cup-shaped organ is called Bowman capsule. Bowman capsule raises and form proximal convoluted tubule, and then proximal convoluted tubule move to the loop of Henle, and loop of Henle move to the distal convoluted tubule. From distal convoluted tubule, urine form and collected into the collecting duct. This is it. After that, the capillary network. The blood capillaries in woman capsule are called glomerulus. Arterioles from renal artery. Okay, and then they come back from the glomerulus and make a network on proximal and distal convoluted tubules. Okay, proximal and distal convoluted tubule have the network of capillaries which is called peritubular capillaries. After that, loop of Henle has another network of capillaries, and this network of capillary is called vasa recta. So three sections of capillaries: glomerulus, peritubular capillaries, and vasa recta. Okay, after that, what is the function of nephron? How urine is formed, or we can say that the regulatory functions of kidney. Here, regulatory functions of kidney comprises of four sections: ultrafiltration, reabsorption, tubular secretion, and countercurrent exchange. What is ultrafiltration? As you know that the blood has most pressure on capillary section. So when blood enters into the glomerulus, it has high pressure. Because of that, from capillary pores, blood is filtered into the woman capsule. Here, the filtrate have two types of section. One is the excretory product, which are not necessarily present in blood, but also some blood cells, some glucose molecule, and some water and salt molecule enter into the woman capsule because of the high pressure. So they will go back. This is ultrafiltration. After that, reabsorption occurs. Reabsorption is actually the active reabsorption of glomerular filtrate occur in the proximal convoluted tubule, as well as it removes water and solute from the tubular fluid and return them to the circulating blood. Means all the important things like glucose, solutes, water, section of red blood cells will go back at this point, reabsorption into the blood, and substances are reabsorbed from the tubule into the peritubular capillaries. So entry from the glomerulus and exit of important material from the peritubular capillaries. Here two types of functions will be performed. One is the both functions will be performed at loop of Henle. Okay, loop of Henle has permeability to water and sodium ions, but impermeable to any type of solute. Descending loop of Henle, this one, after proximal convoluted tubule, this section of the loop of Henle has permeability of sodium and water, and it is all impermeable to all other portions or all other materials. And ascending loop of Henle, the other portion of the loop of Henle, this section is impermeable to water and permeable to other solutes and salts. After that, when water and blood filtrate enter, these arrows show entry and exit of all the material. Like after glomerulus or after woman capsule, in proximal convoluted tubule, reabsorption occur. Means from proximal to capillary. What are the things which go back to the blood? Glucose, amino acid, protein, vitamin, lactate, urea, uric acid. Here, Urea, uric acid, creatinine, and some drugs are not useful for blood in more quantity. So they will again enter into the proximal convoluted tubule. Whole time to the distal convoluted tubule. Exchange and re-exchange will occur. After that, when water is much more, if water is much more in the filtrate, then it will go out and enter into the blood. After that, more urea enter into the filtrate. Why water remove and urea enter into the filtrate? Because urine is always in its concentrated form until and unless you drink much more urine, much more water to make urine diluted. Here, salts, nutrients, ions, and solutes are to excrete out from distal convoluted tubule. Again, water is lost. Again, extra ions and solutes enter into the urine. Here, the filtrate now called at the end of the distal convoluted tubule is called urine. Okay, and then urine again remove extra urea and water, and in its concentrated form move out from the distal convoluted tubule to collecting duct. Now the third section is tubular secretion. What do you mean by tubular secretion? Actually, there are two types of tubes, the proximal convoluted tubule, distal convoluted tubule. These tubes actually secrete some chemicals to produce active transport and passive diffusion to reabsorb the material. Here, like these materials are reabsorbed in the blood because these tubules are porous and help in secretion of these substances. Usually only a few substances are secreted and are typically waste products. This is the tubular secretion, where from tubules to capillary molecules can move. Now the fourth section, which is counter current exchange. Counter current exchange is actually of two types. One counter current is can be termed as counter current multiplier, which occurs in loop of Henle. It helps in the balancing of water and salt in the urine according to the blood, as well as the other counter current help in the section of vasa recta in blood capillaries and provide nutrients. 1% of the blood that flows through the kidney passes through the vasa recta at a very slower speed so that nephron can get nutrients to perform all the function. So two types of counter current exchange are there. Okay. Now, why counter current exchange is important? So here it helps to maintain the hyperosmolarity of medulla while providing blood to renal medulla. Renal medulla is provided blood by counter current exchange. Second is by the vasa recta supplying the long loop of Henle of juxtaglomerular nephron. You do not aware of this term, but the longer loop of Henle is called juxtaglomerular nephron. Okay, as the blood passes through the descending part of vasa recta, it loses water and gains solutes due to increasing osmolarity in medullary interstitium. Reverse happens when the blood returns back through the ascending part of the vasa recta. What is this? This is the process of balancing here. In descending, water remove out. In ascending, ions remove out. This is the thing which is actually counter current exchange or balancing of the body, which we have discussed, which we are discussing here. This one. Thus, hyperosmolarity of medulla remain undisturbed. Okay. Now, which type of nephrons are present in the kidney? As you see in the picture, there are two types of nephron. This is the portion of renal cortex and this is the portion of renal medulla. First nephron have the length of long length and have the area of renal cortex as well as renal medulla. But the second nephron, as you see, do not have loop of Henle much longer and it is only present in the renal cortex. So we can say that there are two types of nephron, cortical nephron and juxtamedullary nephron. 85% of nephrons are cortical and present in the cortex section, and juxtamedullary nephron are closer to the renal medulla and loop of Henle deep into the renal pyramid. So why there are two types of nephron? Because if there are so many juxtamedullary nephron, then water and ions are reabsorbed from the blood and urine will form in much more diluted form. Concentrated form. So there is a balance between the cortex and medulla nephron type of nephron, so that your urine, if one nephron make diluted urine, then other nephron make concentrated urine, so that balance or homeostasis occur in the formation of urine through the kidney. After that, how many hormones are there which can affect the working of kidney? So there are three hormones. One is antidiuretic hormone, the other is aldosterone, and the third one is parathyroid hormone. First, antidiuretic hormone is present in proximal convoluted tubule and all the tubular section of kidney. It helps in the maintenance of water in your body. It checks the blood water level so that urine from urine it can reabsorb water into the blood. After that, aldosterone. Aldosterone is the steroid hormone. Its main role is to regulate salt and water in the body, thus having an effect on blood pressure. So blood solubility or blood hypertoxicity or hypertonicity or hypotonicity is depend on the one hormone, which is aldosterone. After that, the parathyroid. Parathyroid is the hormone which help in the detection or balancing of calcium level. Calcium level in blood is also important, is very much important, and it raises the level of calcium in blood by breaking down the bone so that calcium is released, increasing the body's ability to absorb calcium from food. After that, composition of urine, how much or how many things are present in urine. Most importantly, sodium, potassium, creatine, uric acid, urea, water. These are the major portions, and ammonia are the major portions of urine, but some other salts, magnesium, phosphate, sulfate, chlorides are also present in urine. Why kidney is termed as osmoregulatory as well as excretory organ? So we can say that by performing excretion process, means removal of urine, kidney also balance the toxicity level, solubility level of blood. So we can say that osmoregulatory function is also performed by kidney. This is our lecture today. Thank you.